Enterobacteriaceae, Wikipedia audio. See text. The Enterobacteriaceae are a large family of gram-negative bacteria that includes, along with many harmless symbionts, many of the more familiar pathogens, such as Salmonella, Escherichia coli, Yersinia pestis, Klebsiella, and Shigella. Other disease-causing bacteria in this family include Proteus, Enterobacter, Serratia, and Citrobacter. This family is the only representative in the order Enterobacteriales of the class Gamma Proteobacteria in the phylum Proteobacteria. Phylogenetically, in the Enterobacteriales, several peptidoglycan-less insect endosymbionts form a sister clade to the Enterobacteriaceae, but as they are not validly described, this group is not officially a taxon. Examples of these species are Sodalus, Buchnera, Wigglesworthia, Bomoniaceae delanicola, and Blocomonia, but not former Rickettsias. Members of the Enterobacteriaceae can be trivially referred to as Enterobacteria or enteric bacteria, as several members live in the intestines of animals. In fact, the etymology of the family is Enterobacterium with the suffix to designate a family not after the genus Enterobacter, and the type genus is Escherichia. Members of the Enterobacteriaceae are bacilli, and are typically 1–5 μm in length. They typically appear as medium to large-sized gray colonies on blood agar, although some can express pigments. Like other proteobacteria, Enterobacteriaceae have gram-negative stains, and they are facultative anaerobes, fermenting sugars to produce lactic acid and various other end products. Most also reduce nitrate to nitrite, although exceptions exist. Unlike most similar bacteria, Enterobacteriaceae generally lack cytochrome C oxidase, although there are exceptions. Most have many flagella used to move about, but a few genera are non-modal. They are not spore-forming. Catalase reactions vary among Enterobacteriaceae. Characteristics. Many members of this family are a normal part of the gut flora found in the intestines of humans and other animals, while others are found in water or soil, or are parasites on a variety of different animals and plants. Escherichia coli is one of the most important model organisms, and its genetics and biochemistry have been closely studied. Most members of Enterobacteriaceae have peritrichous, type I fimbriae involved in the adhesion of the bacterial cells to their hosts. Some enterobacteria produce endotoxins. Endotoxins reside in the cell wall and are released when the cell dies and the cell wall disintegrates. Some members of the Enterobacteriaceae produce endotoxins that, when released into the bloodstream following cell lysis, cause a systemic inflammatory and vasodilatory response. The most severe form of this is known as endotoxic shock, which can be rapidly fatal. Phenol red, tryptone broth, phenylalanine agar for detection of production of deaminase, which converts phenylalanine to phenylpyruvic acid. Methyl red or Voges-Proskauer tests depend on the digestion of glucose. The methyl red tests for acid end products. The Voges-Proskauer tests for the production of acetyl methylcarbinol. Catalase test on nutrient agar tests for the production of catalase enzyme, which splits hydrogen peroxide and releases oxygen gas. Oxidase test on nutrient agar tests for the production of the enzyme oxidase, which reacts with an aromatic amine to produce a purple color. Nutrient gelatin tests to detect activity of the enzyme gelatinase. To identify different genera of Enterobacteriaceae, a microbiologist may run a series of tests in the lab. These include In a clinical setting, 3 species make up 80 to 95% of all isolates identified. These are Escherichia coli, Klebsiella pneumoniae, and Proteus mirabilis. Several Enterobacteriaceae strains have been isolated which are resistant to antibiotics, including carbapenems, which are often claimed as the last line of antibiotic defense against resistant organisms. For instance, some Klebsiella pneumoniae strains are carbapenem resistant. Genera. Accepted. Candidatus. Provisional. Identification. Antibiotic resistance.